for kind introduction. Thanks for kind introductions. And hello, everyone. My name is Seong Kim from KAIST in Professor His Labs. Before starting my talk, I would like to appreciate the BioCars communities for giving me an opportunity to share our recent work. Today, I show how the LOV2 domain transitions from its monomeric ground state to the light-induced dimeric photo product. Let me, okay. Before getting into the details, let me start with acknowledgement. This work involves several main contributors who are now former lab members. So today I'll be presenting the result on behalf of them. The main concepts and the design of the project was developed by myself together with Professor Sahi, and sample preparations and initial dynamic studies by Dr. Sori Yoons and Dr. Chang-in Kim, and molecular dynamics by Dr. Sang-jin Lee. And other biochemical experiment, including the mutagenesis and BIM9 experiment, was conducted with all authors. Importantly, this project was started during the pandemics. In other words, many of the initial experiments have to be conducted remotely. So we are very grateful to the BIM9 scientists, especially Dr. Koshileva and Dr. Hanning, for helping us carrying out this experiment. So in this study, we use a time-reserved X-ray liquidography, which is also called as a time-reserved X-ray solution scattering and molecular dynamic to see how a local photochemical event into the global protein associations. Over the next 20 minutes, I will first give you a brief background on the LOV2 domain and then show the Trexel feature and kinetic model and explain the structure of the each intermediate. And finally discuss the dimeric interface and the overall protocycles. As you know that in cell, proteins perform an essential structural and functional roles. And to do this, they often form oligomers and dissociate into monomers. And however, these processes are often very fast and occurs in a complex environment and make them difficult to study at the molecular level. But among these oligomeric transitions, phototropin 1 is an excellent model for studying protein associations. It forms dimer in a light and stay monomeric in a dark, allowing process control with light. And this phototropin is blue light receptor protein kinase in plant with the two LOV domains and kinase. And dimerizations trigger the downstream signaling by the kinase. And importantly, this process is mainly mediated by the LOV2 domain. In the case of LOV domain, previous study have shown that when FMN observed the blue light, it forms a covalent bond, covalent adduct with a conserved the internal system residue. And after forming a photoadduct, a photochemical event occurs on a very fast time scale, and this event initiates a conformation change in a main structure scaffold of the LOV2 domain, and eventually leading to the unfolding of the alpha helix and A-alpha. And our central
Your
question
attention,
that
please. Stored beam has been lost. We're investigating. Repeating. Stored beam has been lost. We're investigating.
then becomes, how does this rapid local formation of the photoadduct propagate into larger scale structural chains that enable LOV to mediate the phototropin's functions? In crystals, LOV2 domains are tightly packed and restricting motions. Thus, light-induced change cannot be fully resolved, and many dynamic intermediates remain hidden. Therefore, the time reserved study at the solution phase are required to understand the mechanism of LOV2 photoreactions. So to capture this dynamic change, we employ the time-reserved X-ray liquidography shortly Trexel, which is also called as a time-reserved X-ray solution scattering, and we expect that Trexel provides an efficient way to observe the light-induced structural change of LOV domain in real time and real solution in solutions. To perform this Trexel experiment, we use biocars, and our X-ray beam has a size of about 30 micron at 12 kb, and we use the nanosecond lasers using wavelengths of 450 nanometers and spot size about 100 microns. And the main sample we investigate were wild type AS LOV2 and I427V mutant, of which photocycles proceeds faster than that of the wild type. After optimizing the experimental conditions, we obtained beautiful structural data capturing the time reserved structure change of LOV2 domain. Next, we analogize the structural data to reveal the LOV2 reaction pathway. So using SVD analysis, we identify three structurally distinct components, and subsequently, we apply the kinetic component analysis to extract the species associated difference scattering curve, shortly size, and this curve provide a direct structural insight into the transition intermediate. For the mutant, I1 forms within 4.62 microseconds, and I2 forms with 130 microseconds, and I3 forms with 3.4 milliseconds. And this sequence show how LOV2 transition from its initial state to the light-induced photoproduct. So people discussing the structure feature of each intermediate, we first examine the structure a SAT curve. So starting with the SAT one correspond to the intermediate one, we see a small positive peak at a small angle. And the low intensity suggested that only a minor structure change occur at this early stage compared to the later intermediate. Moving on to the SAT2, which is correspond to the intermediate 2, we observe a negative peak at a small angle. And the intensity is more than 5 times that of the SAS1, indicating that the intermediate 2 undergoes much larger structure change than the initial intermediate. And this one shows that the conformational rearrangements start to accelerate as the protein progress along the photoreaction pathway. And finally, the SAT3 represents that intermediate 3. Here we see a strong signal at a small angle with both positive and negative peaks. And the intensity is much greater than that of the SAT1 and 2, indicating that the major structure change, particularly during this transition from intermediate 2 to 3. And this confirms that the largest conformational rearrangement occur in the later stage of the photoreaction pathway. So since SAT3 correspond to the intermediate 3 and is the last intermediate in our kinetic model, we predict it to be the final photoproduct. And this prediction was confirmed by static SAX measurement, and the difference between the light irradiated and the ground state scattering curve match SAT3 very closely. Next, we turn to the structural modeling of the LOV2 light-induced state. Our main consideration was as follows. The earliest intermediate resembled the ground state with a folded J-alpha helix, while the later intermediate involved an unfolded J-alpha helix as reported in spectroscopic studies. To explore the possible conformations, we generate the candidate structures using molecular dynamics and non-equilibrium molecular dynamics, allowing unfolding of J-alpha helix along the X, Y, and Z directions from the ground state structures. For each candidate, we calculate theoretical difference scattering curve and compare them to the SAS1 and 2 and 3 using a KAIST scared fittings. And this allowed us select the structure that best represent each intermediate and visualize how LOV2 change during the photocycles. After MD dynamics, we could obtain the model structures of intermediate 1 and 2. And let's look at the structure model for the part 2 intermediate. For I1 correspond to the size 1, we see only a minor local structure change, mainly in the central beta strand and the J-alpha helix. And for I2 correspond to the SAT2, the J-alpha helix is fully unfolding along the plus Y and And plus, these versions best explain the SATU curve. And in the best MD model, the A-alpha hierarchy also unfolded. And this model show how the structure changes grow from such a small local rearrangement in I1 to the larger scale unfolding in intermediate 2. Now, when we examine the intermediate 3, what I mean is for the product correspond to the SAS3, we found something surprising and something wrong. Structural analysis of I3 alone could not be fully explain the experimental data at all. And this unexpected strong signal for I3 indicated that additional larger structural chains occur between I2 and the final photo product. And this means that the transition from I2 to 3 involve more than a simple helix unfolding, and it reflects a major rearrangement that we had not anticipated from earlier spectroscopic studies. So to understand the structure of the final photo product, we turn to the static flux measurement again. And the RD value for the ground state is about 14.75 angstrom, while the light activated state increased to the approximately about 22.7 angstrom. And these large increments indicated that substantial global structure changes in I3, much larger than those seen in I1 or I2, and it cannot be explained by helix unfolding alone. Combined with what is known about the phototropin structure, this ID value suggested that the I3 or LOV2 domain likely adapt dimeric conformations. In other words, the major rearrangement we observe in I3 create the structural basis for the dimerizations and linking the traxel intermediate to the final functional state. So to elucidate the structure of the I3, we explore the possible structure for the light-activated dimers. We consider three types of dimeric interfaces commonly observed in LOV domain. As you can see in a lab, as you can see in a panel denoted as A, we look at three possible interface, the A-alpha helix forming a coil, toil, and the beta scaffold interactions, and a combination where both A-alpha and J-alpha helix form a coil structures. As is shown in a structure in a three rows. For each interface types, we also consider two monomer states. Two monomer state. Both monomers are light activated and shown in the left panel, denoted as A, and which we call the homo light and light LL, or one light activated and the other in a ground state, shown in a panel B, called hetero L and G. By combining this possibility, we generate 6 candidate dimer structures, and using MD simulations, we evaluated to identify the most likely light activated light dimer configurations. After fitting the curve of each candidate with SAS3, we conclude that the beta scaffold interface with both monomers and a light activated state homo LL provide the best fit to the experimental data. And this suggests that in the final photo product, the two monomers interact primarily through the beta scaffold, forming a stable light activated dimer. So to validate our model, we compared the SAS3 and static difference scattering curve and best fit curve of the optimal photo product structures. And we found a good agreement with which support the model derived from Traxel 2. And additionally, low resolution structure reconstructed from static sacks aligned well with this optimal structure too. Here we highlight the key part for the dimerizations. J alpha helix in blue, fully unfolded. A alpha in red, also unfolded, and especially lysine 413 and 475 salt grid form in other spheres. Altogether, this shown that the light-activated dimer formed through the beta scaffold and representing the final stage of the ASLOV2. To validate this proposed dimeric interface, we examine the side exclusion chromatographic profiles of K413E and the E475K mutant, which is designed to disrupt the electrostatic interaction between dimer interface. And we observed that both mutants exhibit a decreased dimer fractions compared to the wild type, even under light activations. And this result supported our model, indicating that the electrostatic interaction between these two residues are critical for stabilizing the light activated dimer. So to further validate our finding, we also collect our tracer data on the wild type sample, which has a slow photocycle compared to the mutant. And the different scattering curve for the wild type are similar to those of mutant, but the transition occur more slowly. And we performed the global kinetic analysis on the wild type data too. And this confirmed that the sequential intermediate and the overall photocycle mechanisms observed in a mutant are consistent with the wild types. And this demonstrates the generality of our model. So in summary, we employ the Trexler to characterize the light-induced structure dynamics of LOV2 domain. And this finding reveals how a local photochemical event can propagate through the protein to drive a light-dependent dimerization, so providing a detailed molecular understanding of the ASLOV2 photocycle. Thank you for your kind of attention.